DNA replication, or the copying of a cell's DNA, is no simple task. There are about 3 billion base pairs of DNA in your genome, all of which must be accurately copied when any one of your trillion of cells divides. The basic mechanisms of DNA replication are similar across organisms. In this, they primarily use the bacterium E. coli and several other bacteria in studying the mechanisms of replication, but do note that these mechanisms are similar to how it happens in eukaryotic cells just like ours. Now, let's take a look at the different proteins and enzymes that carry out the replication and see how they work together to ensure accurate and complete replication of the DNA.
DNA replication starts with the unwinding of the double helix shape of your DNA. We could see it in here in the first image. The DNA is still connected to each other with their bases, but we have here the helicase in gold or blue, is it? Blue gold color. This one would be moving either from 5 prime to 3 prime or 3 prime to 5 prime. And its job is to break the hydrogen bonds in between the bases of your DNA. It will result into something like this. It will look like that. This area in here, where there is a big space, is the replication fork. It is also defined as the area where replication will take place. These two strands that we have produced upon unwinding of the helicase would be the template of the new DNA strands, and it will follow the rules of the semi-conservative replication of DNA. The new strand of this will be adapting the 5 prime to 3 prime direction, and the strand complementing this one will be adapting 3 prime in here and then 5 prime in here. We will see that in the next images. Okay, and these two are not one and the same. The processes in between them are not the same as well. They will have two different mechanisms, and they will have some enzymes that are different or that are not present depending on the strand that we are producing. So let's see the lagging strand and the leading strand. So the leading strand in here would be the one that is continuously adding bases when an enzyme is attached to it. Okay, and we will know that it is the leading strand because the movement of your enzyme or the addition of the bases would be going towards where the replication fork is. Okay, on the other hand, the lagging strand is moving the opposite direction, so it's moving away from the replication fork, and this lagging strand will not be adding fragments or bases in a similar way as a leading strand would be doing. We will highlight that one later on. So, leading strand. For leading strand, the addition of bases will happen from 5 prime to 3 prime. That is the direction it will happen in. This direction also connotes the position of the phosphate groups in your new strand based on semi-conservative and nitrogenous bases pairing. So in here, we can see an orange circle. This one shows you the DNA polymerase. And the job of DNA polymerase would be moving from 5 prime to 3 prime. Yeah, again. It is done in a continuous process, continuous motion towards the fork. It will be adding the nitrogenous bases continuously until it reaches the end of the fork where the replication will be stopping. Next, lagging strand. For lagging strand, DNA polymerase would still be involved, but before the DNA polymerase would do its job, there is another enzyme that would have to participate first. We call this enzyme as primase. It is shown in this pink gold color. And the job of this DNA primase is to attach to the parent DNA strand, and then this one will be attaching an RNA primer. Okay, this is the RNA primer. The DNA primer would be the starting point, or it could be part of the fragment. That the first enzyme that will be involved in the lagging strand is this pink one. We call this one as a primase, and the job of a primase is to add an RNA primer, which I am pointing in here. Once this one is added, this RNA primase will be moving away. Next, the DNA polymerase can now start adding fragments of DNA. This one is done in the 5' to 3' direction. It will be forming the fragments that we call as Okazaki fragments, and we could find it in here. These Okazaki fragments are not RNA, they are DNA-based sequences. Okay? Then, this process will continue on and on and on, and this one will also continue on and on and on until all of the gaps are filled up, just like in here. Once these are all now complete, we still have to remember that the primers, or the RNA primers that were added, are not the same as the DNA base pairs, because RNA have uracil, DNA doesn't have uracil. Therefore, these primers would have to be replaced again with DNA bases. That's right, I know you said it in your brain. So what would it do in order for this this RNA be replaced with DNA base sequences? This pink blue blob in here, or circle, I keep saying blob, shows us exonuclease. The job of exonuclease is to attach to the RNA primer, and it will be released. Okay, once these two are released, a new DNA polymerase, a DNA polymerase will be attaching, and this one will be guiding new DNA bases into this section of the DNA, which will result into something like this. We could see now that the former RNA primers are now part of the, or are of the same sequence as a DNA. This will be done again and again and again until all primers are removed and replaced with DNA bases. After everything has been replaced, we have to take note that there are spaces in between, or there are gaps in between the segments or the fragments itself. Therefore, this color gold circle in here shows us DNA ligase, which will function in joining the fragments by synthesizing phosphodiester bonds in between. It will run through the length of the DNA to check if there are areas that are not sealed. If there are areas that are not sealed or not connected to each other, the DNA ligase will act so that phosphodiester bond will be added to it. Okay, so once this process is done, your replication is now complete. One thing to note though, DNA replication will not happen at one point or one area in the DNA only. It can happen in several areas at the same time, because if you would be doing it from the starting point to the ending point, like literally like opening a zipper up, it will take at least, they say, 1 month in order to finish copying a replication. So in order to prevent this from happening, and be able to complete one cell cycle in at least 24 to 26 hours. What the DNA will be doing is that it will unwind several areas up, but not the whole thing all at the same time.